name is Dominic Boceck, and today I want to present you aiDIVA, which stands for Augmented Intelligence Disease Variant Analysis. aiDIVA is a tool that we developed to aid clinicians in the disease variant analysis. So the diagnosis of rare Mendelian diseases is very challenging, and less than 40% of the cases can be solved using exome or genome sequencing. And there are two main difficulties because why that is the case. First, the accurate pathogenicity classification of all the detected variants that we get from the exome or genome sequencing. And secondly, how to prioritize all these variants to find the causal variant among these thousands of variants. To help our clinicians with the diagnosis of rare diseases, we created aiDIVA, which is meant to be a clinical decision support system. Here on the right side, I have the workflow of aiDIVA. aiDIVA is structured into three parts. The annotation part also includes a pre-filtering step and annotates all the genomic features that are used in the following steps of aiDIVA. Then we have the pathogenicity scoring part and the prioritization part. So we start with the patient VCF file, then we filter this and annotate all the genomic features. Then afterwards, we score we classify the pathogenicity of all the variants using, for the ISMB variants and in-frame indel variants, we use custom random forest models. And for frameshift variants, we use predefined pathogenicity scores, and for splicing variants, we use the DBSC SNV database that has predefined pathogenicity scores for splicing variants. So our two random forest models were trained on pathogenic and benign variants from ClinVar. ClinVar is a database with information about the pathogenicity of variants that cause diseases. If we have all these pathogenicity scores, we combine these scores, these classification scores, with the phenotype of the patient that was reported from the clinician. The phenotype is reported as HBO terms, where each HBO term stands for a single phenotype that can be observed from the patient. And we also can include clinical knowledge from databases such as ClinVar or other databases that has information about clinical relevance of a variant. So all these scores are combined in a weighted approach, which was trained on our benchmark samples, which I will talk on the next slide. So in the end, finally, we will get a causal variant ranked list, where ideally we can find the causal variant on the top one position. So to show the performance of aiDIVA, we created a benchmark study consisting of in-house data of clinical diagnosed patients. All these patients are suffering from a rare Mendelian disease, which is usually caused by one or two single genomic variants. Our benchmark set consists of a total of 3,328 samples with one or two clear causal variants. And all these causal variants were identified by exome or genome sequencing. And finally, we also compared aiDIVA to Exomizer, which is a widely used tool for exome-based rare disease diagnostics. So now to the results. Here on the left, we have violin plot showing the rank of the causal variant where the causal variant was ranked with aiDIVA and Exomizer. So here on the top, so ideally here on the top, here's the rank 1, so 1 position. So ideally we want all the causal variants at the top 1 position. we say the range from 1 to 10 is very good. The range from 10 to 25 is still okay. And if the rank was above 25, we say it didn't work so well. So in comparison, we can clearly see that aiDIVA outperforms ECCB on our benchmark set. And even the variants that for the samples for which the prioritization didn't work well also are less with aiDIVA than with ECCB. So here on the right, we have a table showing the same results as percentages. So in over 40% of the cases, aiDIVA ranks the causal variant in the top 1 position. And over 70% we get in the top 5 position. And in the well-performing range with top 10, we get 85%. And if we also include the still well-working range, we come over 90% of the cases. So to further improve aiDIVA, we try to analyze the variants that ranked below or above 25, so that for which it didn't work well. And we want to try to extend the range of supported variants to the non-coding part of the genome. aiDIVA is fully implemented in Python 3 under the MIT license and can be found on GitHub under the following link. Thank you for your attention.